An overview of Mendelian genetics. Prior to the mid-1800s, there were many different ideas about how traits are passed on from parents to offspring. But it was Gregor Mendel who, through his pea plant experiments, actually discovered the basic principles of heredity. He deduced that genes come in pairs and are inherited as distinct units, one from each parent. He tracked the segregation of parental genes and their appearance in offspring as dominant or recessive traits. And he recognized the mathematical patterns of inheritance from one generation to the next. Mendel was able to cross true breeding parents and analyze their offspring. Then, by self-fertilizing those offspring, he determined that the offspring inherit one gene allele from each parent when the sex cells unite in fertilization. The outward appearance of the traits are known as the phenotype. And Mendel recognized that the only way that the first generation, or F1 offspring, would exhibit the same phenotype of only one of the parents, such as the round peas, but that the second generation would exhibit a 3/4 to 1/4 phenotypic ratio, would be if each trait had two distinct variant alleles, and that a parent provides only one allele to the offspring through the gametes. Therefore, each parent must be homozygous for the different alleles. From this, he recognized then that the F1 must be heterozygous, or have two different alleles, and that the one is completely dominant over the other. So in the case of the round versus the wrinkled, the heterozygote would appear round, even though they were a carrier of the wrinkled allele. Mendel realized that the genotype is the actual allelic makeup of the individual. So he described those with the 3/4 ratio parent as, again, the dominant trait, and the lower frequency as the recessive, so that those that were 1/4 would be homozygote recessive. And this is why we see this 3-to-1 ratio. 3/4 of the individuals in the F2 carry the dominant allele and therefore exhibit the round phenotype, whereas 1/4 have both of the recessive alleles. They're homozygous recessive, and thus are wrinkled in appearance. Mendel observed the same pattern for all of the 7 pea plant traits that he examined. Again, for example, the parent generation of a cross between purple flowers and white flowers produced an F1 generation of all purple flowers. So therefore, the purple flowers of the F1 had to be heterozygous. These purple flowers were self-crossed, again producing a 3/4 to 1/4 ratio of purple to white flowers. He called the F1 crosses monohybrid crosses because he, again, recognized that the trait was governed by just one gene with those two alleles, and that those F1s were heterozygous, being hybrids. So when you cross two heterozygotes in a monohybrid cross, you see this 3/4 to 1/4 phenotypic ratio. So what he understood here was that the alleles must be segregating equally during gamete formation. So this is how he came up with his principle known as equal segregation. Mendel went a step further and looked at 2 traits. He crossed a parent that had yellow and round peas by one that had green and wrinkled peas. The F1 generation exhibited the dominant phenotype for both traits. So the F1 were all yellow and round, again supporting the principle of equal segregation. The F1s were then self-crossed, and again, what we see here now, because it's two traits, the F1 by F1 is called a dihybrid cross. The result now was a 9 to 3 to 3 to 1 ratio. So that, again, 9/16 of all the plants were dominant for both of the traits, thus 9/16 were yellow and round. 3/16 were dominant for one trait but recessive for the other, such as yellow and wrinkled, right? Or green and round. And then 1/16 were recessive for both, thus they were green and wrinkled. From these results, Mendel was able to conclude that the two alleles for each gene segregate independently of one another. Genes for different traits are sorted independently from one another so that the inheritance of one trait is not dependent on the inheritance of another. This is called Mendel's principle of independent assortment. So what we can see here is Mendelian genetics describes how some traits are determined by a single gene and that they exhibit complete dominance over the recessive allele. The genes for two different traits will assort independent for one another. Thus, you can predict the outcomes of crosses if you know whether or not a trait is dominant or recessive. And if you don't know if a trait is dominant or recessive, through the crosses, you should be able to figure this out, looking at the ratios. So again, a monohybrid cross should produce a 3 to 1 ratio. A dihybrid cross should produce a 9 to 3 to 3 to 1 ratio. If an individual is exhibiting a dominant phenotype, but you don't know if that individual is heterozygous or homozygous dominant, it can be crossed with a known homozygote recessive. This is called a test cross. And by examining the offspring, you can figure out what the genotype of that parent was. So if an individual is crossed with a known homozygous recessive and half of the offspring are homozygous recessive, then you know that that parent had to be a heterozygote. Whereas if all of the offspring exhibit just the dominant phenotype, then you can deduce that parent was homozygote dominant. A lot of genetic disorders follow Mendelian patterns of inheritance. For example, homeochromatosis is inherited and it is caused by a recessive allele. If two partners are both heterozygous, what is the probability that they will have an infected offspring? Again, we're looking at a single gene trait, and we already know that it follows Mendelian rules. Therefore, we can expect a probability of 1 out of 4 offspring having the condition. To predict the probability of 2 of their children having the disease, you would just take the independent probability of one event and multiply it by the independent probability of another. Therefore, it would be 1/4 times 1/4. This equals a probability of 1/16 that 2 of their offspring would be affected with the disorder. So trying to understand whether or not a human health disorder is dominant or recessive is not always just as simple as looking at crosses like Mendel did, because the sample size in human families is much too low. Also, the generation time is very long. Therefore, geneticists use an important tool known as a pedigree. Pedigrees allow them to understand the inheritance pattern and then make accurate predictions about the probability of passing on traits. Each pedigree traces a trait through the generations of the same family. An important application of the pedigree is to calculate the probability that a future child will have a particular genotype or phenotype. However, many traits are much more complicated than simple Mendelian traits. This is because they either don't exhibit complete dominance, or there are multiple alleles, or more than one gene is responsible for the trait. So when you are studying, it's important that you recognize some of these non-Mendelian patterns, such as incomplete dominance, codominance, multiple alleles, and multiple genes. And keep in mind that many traits are affected by the environment. These are known as quantitative traits and can be very difficult to study and understand. So the genetic experiments that Mendel did with his pea plants took him 8 years, and he finished that in 1865 was the publication. It wasn't until 1910 that Thomas Hunt Morgan was able to use Drosophila to actually confirm that genes are located on chromosomes. So it was no small feat that Mendel was able to come up with his principles without the actual knowledge of chromosomes, but through his understanding of mathematics and probability, he was able to figure it out. So it's a pretty amazing feat. I hope this helps you understand genetics of Mendel a little bit better. Thanks. Have a good day.